The brain is the most complex part of the human body. One way of unlocking the secrets is by reverse engineering the brain. Dr. Sergiu Pasca has made it his life's work to understand how the brain builds itself. Dr. Pasca is a professor of psychiatry and behavioral sciences at Stanford University.
The brain is a remarkable organ, and the human brain in particular is really incredible. We have more than 86 billion neurons and probably 86 billion glial cells that are supporting it. The problem is that the human brain is to a large extent inaccessible. It's inaccessible in the adult, but it's inaccessible during its development. And that has really precluded our understanding of how the human brain becomes the human brain and how processes that assemble it go awry in diseases. And that's probably one of the main reasons why we have such a poor understanding of psychiatric disorders today.
Dr. Pasca's lab is trying to understand the underlying mechanisms that lead to neuropsychiatric diseases, including autism and schizophrenia.
We're looking at a cortex on the side, and then this is the thalamus. And so
they've done it by developing a new technique to grow simple human brain circuits using stem cells.
Projections all over the place and then releasing neurotransmitters.
Let me ask you about your journey, because setting out in life, this really wasn't a thing, and now here you are. How do you get from a kid to here? Talk to us about your journey.
I think I was interested in doing science very early on. I grew up in Romania during the last few years of communism, but wanted to do science very early on. So I built like my first lab in the basement of my parents' house when I was 10 or 11 and started studying plants and seeing the effect of various chemicals. And then when I went to medical school a few years later, seeing the first patient with autism is really what triggered my interest in the brain. And I realized that is the final frontier.
The research team takes skin cells and reprograms them to essentially push them back in time so they act like stem cells. Scientists then guide these stem cells to develop into different kinds of brain cells called organoids. Organoids can then lump together into a simple human brain circuit called an assembloid.
The secret to this process is careful guidance. In the end, they look like this: tiny clusters of brain cells at the bottom of a dish. And let me be clear, these are not brains in a jar. These are parts of the nervous system in a laboratory dish.
So it's this mystery, it's a secret in many respects. But you've been able to basically unlock that in a sense. I have a statement that I make about the news business, as a journalist, you go out there, this is what you think the story is, but then you get there and it's something entirely different. I want to ask you about the brain. Did you go in with this set of beliefs, this is the way it is, and then you get in and you look at this Petri dish and you're like, wow, it's a different story altogether, or was it pretty much squared?
Oh, it was, a combination of, I guess, wanting to achieve like making human cells and then being a little bit naive about like how complicated the process will be. We thought it would be just a couple of years and it will work out. And of course, it turns out to be almost a decade of actually trying to make the cells at the bottom of a dish. And of course, even in the beginning, when we were making very simple cells sitting flat at the bottom of a Petri dish, which is not obviously what the human brain is. And over the past few years, our dish preparation became ever more complex. Initially, we were putting them at the bottom of a dish. Then we started putting them in three-dimensional cultures. Then we started to assemble them 2x2 or 3x3 to start to form circuits. So it's really been a process of trying to recapitulate the complexity of the human brain.
In your TED Talk, it's fascinating to watch. Here they are, and then they move. I'm not doing a very good job of it, but you know what I'm saying. And then you have, to put it in layman's terms, what we would look at, it's like one blob here, one blob here, one blob here, but it's obviously much more than that. And the connective tissue and the firing, all of it's just so fascinating.
What is remarkable about the brain is that it's not a homogeneous organ. Now, unlike the liver, where almost any single part could play the role of the other part, in the human brain, we have so many regions and nuclei that are connecting with each other. And that's how the remarkable properties of the human brain arise. So initially, we were only able to make actually the cerebral cortex. But the cerebral cortex has multiple types of neurons. It has some that are excitatory and some that are inhibitory. And there's a very delicate balance between these two cell types. And as you can imagine, if you have too much excitation, you can get epilepsy. If you have too little, you can get other conditions, psychiatric conditions. But most of those inhibitory cells are not born in that place of the nervous system. They're actually born very, very far away. And then they literally have to migrate over many, many months up to the second year of life in humans to find their final position. So what we did is we made those two brain regions separately, the one that has all the excitatory cells and the one that has all the inhibitory cells. And then we just put them together, not knowing what would actually happen. And what was surprising for us, and I guess we keep being surprised by the remarkable self-assembly and self-organization of this, the inhibitory neurons knew exactly where to go. You know, being trained as a physician, I very often thought that, you know, the human body is a machine. If you really want to understand it, you need to understand every single part. And if you want to rebuild it or reconstruct it in any way, you need to know exactly how to do that. What we've been learning is that if you know how to make some of these brain regions, they come with the instructions. And so you can, to a large extent, reverse engineer how they're doing it by just putting them in a dish and letting them interact. And that has been really part of this process of, to a large extent, trying to demystify the human brain and hopefully ultimately demystify psychiatric disorders.
When you first started doing this kind of work, it seems like science fiction. It's not possible. Did you run up against some obstacles as a result? I mean, how does one come up with an idea like this?
For sure. I mean, this has been a very long journey. First of all, because for decades, we've been unable to even grow stem cells in the dish. And that became possible in the late 80s. Then, of course, it wasn't possible to access those stem cells from anybody, right? Unless you were to use embryonic stem cells. And then the discovery of this cellular reprogramming, of pushing them back in time, was really transformative and suddenly enabled us to make the stem cells from any individual and then to guide them in a dish. And I was just finishing my clinical studies at that time. I was very interested in studying autism. And of course, it was very frustrating because unlike in any other branch of medicine, we couldn't access the organ of interest. So we would study the mouse brain, or I was studying the cat visual cortex. And of course, it was very frustrating. How are we ever going to get access to human neurons from patients to understand what goes wrong? And so this discovery of being able to turn skin cells into stem cells and then those stem cells into neurons, became clear as one avenue towards like achieving that goal. So I, to a large extent, changed my career track and decided that I will be focusing my work primarily on basic science and try to build models that would allow us to study both how the human brain assembles itself, but also how that goes awry in disease.
Nearly 5.5 million Americans are living with some form of autism spectrum disorder. 1 in 36 children have autism, according to the U.S. Centers for Disease Control and Prevention. The severity of the signs and symptoms vary, but typically include problems with communicating, learning, moving, or paying attention. Timothy syndrome is a rare, life-threatening genetic disorder. In 2022, Dr. Pasca and his team transplanted human organoids from both healthy and Timothy syndrome subjects into rat brains to study differences in brain development. This year, they developed a therapeutic strategy that might correct the defects in Timothy syndrome neurons. This strategy could also apply to other brain development disorders like autism, which was once considered untreatable.
For a long time, it was clear that many psychiatric disorders have a heritable component. We knew that they were running in families, but we didn't know what genes were contributing. And so now, for instance, for autism, we have hundreds of genes that we know are associated with this condition. What this means is that now we can actually take those genes and, take them out, delete them from cells in a dish and see how is that impairing development of the nervous system. And I think the promise is really that by ultimately manipulating the cells and leveraging human genetics and this in vitro models, this in a dish models, we'll be able to understand the biology of these conditions. And we've been making great progress, especially for some forms of autism and epilepsy, again, that are genetically defined, that are caused by specific mutations, and now to a point where we have a very good understanding of how they're actually impairing the nervous system and its development.
So great progress then leads to, once you have a better understanding, you can target remedies then.
Exactly. But one of the challenges has been that, everything that we do is in a dish. And so you never really know whether that would translate in a living organism. Would that defect still be present? Would it be compensated for? You could say, very often we joke saying that in vivo veritas, meaning that in vivo, like in a living organism, there is actually truth, not just what we see in a dish. So what we've been doing over the past 8 years or so is to develop a platform that allows us to also verify that what we've actually identified in a dish would actually translate to in vivo changes or in an organism. And that's where we've developed this transplantation platform, where we can now take some of this 3-dimensional organoid cultures that we can develop in a dish and actually put them in a living rat early in development, so that the human cells integrate into the rat and can even receive sensory input. So you can move the whiskers of the rat and human cells will respond, or the human cells will participate to the behavior of the rat. The promise of this is that now you can transplant cells from patients, neurons from a patient with a specific form of autism, and then, for instance, inject a drug of interest into the rat, but look at the effect on human cells, which is certainly very much needed, because we need to take as many precautions as we can before we would inject any of these drugs into human patients.
Tell us what your dream looks like for the future.
Well, I ever since I saw my first patient with autism, when I was in medical school, and it becomes so clear so quickly that some of these patients, many of whom have intellectual disability as well, it becomes so clear that there's something that we need to understand about the human brain, that those circuits are somehow miswired. There must be a way of understanding. And that frustration, stays on for a very long time. And especially, in my interactions with families, some of these patients have like 60 seizures a day. That's not an easy thing to deal with as a family. And in fact, we see the impact on the parents, on the siblings for most of these families. And of course, autism is a spectrum that we see in the population. We're referring primarily to patients who really have severe forms of autism and intellectual disability, where there is significant suffering. And so my dream is that we will start to understand how some of these behaviorally defined conditions, these psychiatric conditions, arise at the level of circuits and cells. And hopefully we'll find ways of at least improving, if not curing, some of these conditions.
It sounds fantastic, but Dr. Pasca, if I'm a parent of that kid with autism and the life you just described, I want that answer today. How do they know about the research you're doing, and are you there yet? How do you kind of temper their expectations?
Yeah, I think I try to always temper the expectations, because everything takes so much longer than we've ever planned for, and I think that's true not just for psychiatry, but for many other branches of medicine. I think the average time for developing a biologically inspired therapeutic is 25 years. So it's at least 2 decades of work until you actually have it. And I think that's important. And, interestingly, many of the parents of the patients that we work with understand that really well. Many of them already know that maybe this may not have an impact for their children, but they know that they may have for others in the community, or for the others that are going to get diagnosed in the next decade. And that's really remarkable, just to see when sometimes some of these parents will travel from far away to bring their kids here to Stanford so we can take a skin biopsy. Very often not trivial procedure, or not a trivial travel for many of them to do. And I think that speaks to the hope that some of these parents have, if not for their children, for the others in the community.
Well, it's been a great discussion. Thank you so much.
Thank you so much for having me.